Well, I work in a research unit in the University of Nantes in France, in the west of France. And our interest is to try and understand the biology which is responsible for changing the level of isotopes in natural products, in biochemicals, so the chemicals that make up our body, the plants, and everything around us. And so we are trying to understand the factors within different metabolic systems, within different organisms, within bacteria, yeast, animals, and plants. And when you look at the chemicals that make up these living organisms, the isotopes of hydrogen, of carbon, oxygen, nitrogen do not have the same level in every compound. They're what we call fractionated. And this is because the enzymes, the special protein catalysts that make compounds, that make the pathways, have sensitivity to isotopes, to heavy isotopes. And the sensitivity is different for different enzymes, and it's different for different chemicals, and it's different for different carbon or hydrogen positions within the chemical. So if you have a ring, maybe one here is sensitive and one here is not. So this causes a non-statistical distribution, and we want to understand what biochemical and biological and physical chemistry is underlying what we observe. So we make measurements on compounds, and we use two techniques. We use the technique of nuclear magnetic resonance spectroscopy, and this allows us to look at individual positions. And if we want to just look at the whole molecule in one together, then we use a technique of mass spectrometry, which is called isotope ratio mass spectrometry. Now, these two techniques are complementary because for the nuclear magnetic resonance technique, we need a lot of material. We need maybe half a gram of pure chemical. So you cannot always have half a gram. But when we can, we get a lot more information. Now, this may sound very scientific and useless, but it does actually have application. And in fact, the group in Nantes started working in this area in 1980, and I joined them in 1995. And the group was established by Professor Martin with the objective of trying to find a way of measuring that people had added sugar into grape juice when they were making wine. This is a process called chaptalization, and it's illegal. In France, it's illegal. The idea was that sugar that came from a grape and sugar that came from a beetroot or from cane sugar would not have the same isotopic composition. And so if you mix them together, you get a value for your isotope which is not the same as it should be in the natural grape product. This is true, and the method was patented and is now a big European method which is used for all European wines, including Hungarian wines. And the main place that is done is in the north of Italy, in Ispra, in the European Union Research Center there. So what happens is that because you have different plants making the sugars that are fermented, you get different isotope values, especially for deuterium, but also it depends on where it grows. So if you take a wine from Hungary and a wine from Bordeaux, the value will not be the same, even though it's all grape and it's all wine, because there's a geographic effect as well. And we've been hearing today about how water in Nantes has a value of about 150 units, and here it's only about 140. So that's enough to say, well, the wine from Hungary would have a different value in its deuterium isotope from the wine in France. And also, when it rains, you get a different value from when it's very dry. So a very dry year and a very wet year from the same vineyard is not the same. So that's the application, and we do it for wine. We also can do it for olive oil. A very big application is for fruit juice. Fruit juices, the water in the fruit juice especially, some fruit juices are concentrated, shipped concentrated and then diluted. That is perfectly legal as long as it says so on the label. What it mustn't do is say that this is whole fruit juice. So a fruit juice that has been diluted can be sold illegally as whole fruit juice. Again, you can tell that water has been added because it's not the water from Florida or wherever the oranges were being grown. Deuterium depleted water, I have no experience myself of working with deuterium depleted water, but it's very clear that there are significant effects of deuterium depletion. This does not surprise me at all. I work a lot with these catalysts, the enzymes, and these enzymes, as I say, are very sensitive to deuterium. So if you have an enzyme which has water as a substrate, for example, as many enzymes do, it is also sensitive to deuterium in the water. So if you have less deuterium, just like if you have more deuterium, you will get a different rate of reaction. So I can quite understand that when you put many reactions together and you have less deuterium, this will give an accumulated effect, of course, and that you will see changes in metabolism. Certainly, yes, yes. Well, I think during the conference we've certainly seen one or two potential applications of using deuterium depleted water in terms of changing what I'd call undesirable metabolism. That's to say cells which have lost the regulation and are cancerous, tumorous. It does appear that deuterium depleted water may have some impact on that metabolism, perhaps helps to bring it back under regulation. In terms of my own research, I think it would be a research tool to try and understand, again, these factors, what are the important factors in regulating how metabolites are transformed and the rate at which they're transformed, which is also very important.